Hello children, how are you? You must have read the first chapter what we studied yesterday. So today let us start with the second chapter, that is sexual reproduction in flowering plants. So flowers are the wonderful part of the nature. You see lot of attractive flowers. So actually the flowers mainly that is the reproductive part of the plant and helps in pollination. So if there is no flower, the maintenance of the particular species won't be there. It is the main purpose of the flower. But apart from that, other uses of the flower also there. It is source of honey, we get medicine from that. It is mainly, it gives beauty to the nature and surrounding. We get perfume, scents, we use for various decorations. So various purposes we use. Mainly let us go to the structure of flower and the function of flower. So when you see in the case of flower, so mainly when the flower is produced in the plant. The plant when it grows, when during juvenile phase, that is mainly the vegetative phase, what you call in the case of plants. So in the case of vegetative phase, they are not able to produce flower. They will be under growth condition. When it attains the reproductive maturity, the hormonal and structural changes occur in the plant and it results in the development of flower. So when you see the other portion, you know this is sepal, this is petal. Actually petals, they give attractiveness to the flower. It is helpful for pollination. It attracts the pollinators. It may be insects or birds and helps in pollination. Sepals, it protects the flower when it is in the form of bud. Main reproductive part of the flower means that is androecium and gynoecium. So what is androecium? Androecium is a whorl of stamens. Can you see? This is stamen. In the case of stamen, that is no male sex organ. This is filament and this is anther. And gynoecium, it is the female reproductive portion. What is seen in the center, it contains ovum and it develops into ovary. Within the ovary, the seeds will be there. So after fertilization, that develops into fruit. The ovary develops into fruit. Androecium and gynoecium, that is the main reproductive part of the plant. So now let us see the different type of events that is occurring during pre-fertilization. Pre-fertilization means it is preparing for fertilization. That is the development of here pollen grain. We are going to see first step. So that is stamen. Mainly in the stamen, the different structure stamen, microsporangium and pollen grain. How do they what are the development of pollen grain? Let us see. So here when you see in the case of stamen, what will be the stamen? It consists of filament and the anther. Okay, the filament, it is no giving attachment of the anther along with the flower. When you see that anther, it is bilobed in nature. When you see in typical angiosperm, it is bilobed in nature. That you call that as dithecous. Inside the sporangium, inside the anther, there will be sporangium, that you call that as microsporangia. Why do you call that as microsporangia? Because it forms microspores. Microspores are nothing, they are pollen grains. As pollen grains are developed, you call them, that is pollen grains, that is microspores are developed from here. You call that as microsporangium. Okay. So now let us see the structure of microsporangium. So there will be totally, when you see 4 microsporangium will be there. You can see 1, 2, 3, 4 microsporangium will be there. Here 1 microsporangium is fully enlarged, and let us see the structure of one single microsporangium. Each microsporangium, when you see, they are having 4 wall layers. The outermost layer you call that as epidermis, and next to epidermis there will be endothecium, then there will be middle layers, then the innermost layer you call that as tapetum. So here the tapetum, it provides the nutrients for the developing spores. Mainly it is helpful for supplying nutrients. It nourishes the developing pollen grain. Okay. So whatever, then you see the center of microsporangium, there will be sporogenous tissue. Can you see sporogenous tissue? So here it will be sporogenous tissue. The sporogenous tissue is helpful in the development of pollen grains. So this is the matured anther. So when the sporogenous tissue develops into pollen grain, it dehisces, breaks and liberates the pollen grains outside. When it is immature, it is intact. When it is mature, it breaks and liberates the pollen grains. So now let us see the formation of pollen grain. Formation of pollen grain. Pollen grains are microspores. So formation of pollen grain, you call that as microsporogenesis. Microsporogenesis means, already told you, it is formation. As it is helpful in the development of microspore, you call that as microsporogenesis. Microspores are formed from the cell, you call that as microspore mother cell. Where is microspore mother cell present? It is present in sporogenous tissue. The sporogenous tissue of Microsporangium that differentiates into microspore mother cells, otherwise you can call that as pollen mother cell. Then it undergoes meiosis. You know, at the end of meiosis there will be 4 daughter cells are formed. So at the what is formed now, you call that as microspore tetrad. As there are 4 cells are there, you call that as microspore tetrad. Then later, on dehydration, that is removal of water, then slowly it dries up and all the 4 microspores get separated. Okay, and each microspore, it develops into pollen grain. Means from a single microspore tetrad, how many pollen grains will be formed? 4 pollen grains will be formed. 4 microspores results in the formation of 4 pollen grains. Now we see the structure of pollen grain. So when you see, this is the microspore tetrad. So actually this is how the sporogenous mother cell, and slowly it undergoes division and results in the formation of pollen grain. So now let us see the matured pollen grain. This is the matured pollen grain. So what is the structure of the matured pollen grain? So pollen grain, that you call them as, they are the male gametophytes. Okay. So when you see, it is 2 layered. You can see the 2 layers, inner layer and outer layer. So inner layer you call that as intine, and outer layer you can call that as exine. The exine, the outer layer, it is made up of sporopollenin. Okay, it is the sporopollenin, it is the tough layer. It is strong, it can withstand adverse conditions. Okay, because of that, no, the pollen grain won't be degraded when it is taken from the flower. When it is moving by means of for pollination, it won't be getting degraded. Okay, the sporopollenin, it is highly resistant to organic materials. No, it can withstand temperature, strong acids, alkali. Okay, it can live for longer time. Then the exine, it contains one pore, that you call that as, that will be germ pore. And in germ pore, the sporopollenin will be absent. The inner wall, you call that as intine. It is made up of cellulose and pectin. On maturity, the pollen grain, it contains 2 cells. Now the two, actually it was a single cell, but later it, by means of mitosis, it divides into 2 cells. One is bigger and smaller. Why it is bigger and smaller? So here there will be unequal cytoplasmic division. When unequal cytoplasmic division is there, one cell will be bigger and other cell will be small. The upper cell you call that as the bigger cell, vegetative cell, and this cell you call that as generative cell. The vegetative cell is bigger and it contains lot of food reserve. Okay, so then the generative cell is smaller. When you see in the 60% of angiosperms, so pollen grains are shed, they are released during the 2-celled stage. But in some of the angiosperms, what happens? The generative cell, it divides mitotically once again, and it results in the formation of 2 male gametes, and it will be liberated in 3-cell stage. So some 2-cell and in others in 3-cell. How 3-cell? Because the generative cell, once again it divides by means of mitosis and liberated. Okay, so today we will stop this chapter up to this. Then we can continue with the next thing.